Which of the following genetic processes is primarily responsible for creating genetic diversity within human populations? A. Gene duplication. B. Independent assortment. C. DNA methylation. D. Transcription. The correct answer is B. Independent assortment during meiosis allows different combinations of maternal and paternal chromosomes, creating genetic diversity in offspring. In human genetics, what is the primary consequence of a mutation in a somatic cell gene compared to a germline mutation? A. Only germline mutations affect offspring. B. Somatic mutations are inherited through the mitochondrial DNA. C. Somatic mutations can lead to evolutionary changes. D. Germline mutations have no impact on inheritance patterns. The correct answer is A. Somatic mutations occur in non-reproductive cells and are not passed to offspring, whereas germline mutations can be inherited. How does the phenomenon of epistasis affect the expression of genes in human genetics? A. It involves environmental factors overriding genetic factors. B. It results in codominance between alleles. C. One gene can mask or alter the expression of another gene. D. It leads to mutations in non-coding DNA regions. The correct answer is C. Epistasis occurs when the expression of one gene is masked or modified by another gene, affecting phenotypic outcomes. What is the key characteristic of a heterozygous individual in terms of allelic composition? A. Two identical alleles at a locus. B. Two different alleles at a locus. C. A deletion in one allele. D. A duplication in both alleles. The correct answer is B. A heterozygous individual has two different alleles for a specific gene at a given locus. In the context of human mitochondrial inheritance, which of the following statements is correct? A. Mitochondrial DNA is inherited from both parents. B. Mitochondrial DNA is only passed from mother to offspring. C. Mitochondrial DNA is encoded in the nucleus. D. Mitochondrial inheritance follows an autosomal pattern. The correct answer is B. Mitochondrial DNA is maternally inherited, meaning it is passed exclusively from mother to offspring. Which of the following best describes the role of a telomere in human chromosomes? A. To encode genetic information. B. To initiate cell division. C. To protect chromosomes from degradation. D. To facilitate transcription. The correct answer is C. Telomeres protect chromosome ends from deterioration or fusion with neighboring chromosomes, maintaining chromosome stability. What is the significance of single nucleotide polymorphisms, SNPs, in human genetics? A. They lead to chromosomal translocations. B. They are key markers for genetic variation within populations. C. They cause gene duplication. D. They are the main cause of mitochondrial mutations. The correct answer is B. SNPs are single base pair variations in DNA that are important for studying genetic diversity and identifying disease-linked variants. Which enzyme is responsible for adding new nucleotides during DNA replication in human cells? A. DNA helicase. B. RNA polymerase. C. DNA polymerase. D. Ligase. The correct answer is C. DNA polymerase is the enzyme that synthesizes new DNA strands by adding nucleotides complementary to the template strand during replication. In human genetic disorders, which term describes a mutation that causes a premature stop codon, resulting in a truncated protein? A. Missense mutation. B. Nonsense mutation. C. Silent mutation. D. Frameshift mutation. The correct answer is B. A nonsense mutation introduces a premature stop codon, which truncates protein synthesis and often results in a non-functional protein. Which of the following chromosomal abnormalities is specifically associated with Down syndrome? A. Trisomy 21. B. Trisomy 18. C. Monosomy X. D. Triploidy. The correct answer is A. Down syndrome is caused by trisomy 21, where an individual has 3 copies of chromosome 21 instead of the usual 2. Which term refers to the genetic mechanism that allows a cell to turn off one X chromosome in females? A. Genomic imprinting. B. X inactivation. C. Gene silencing. D. Epigenetic marking. The correct answer is B. X inactivation is a process by which one of the 2 X chromosomes in females is randomly inactivated, balancing gene dosage between males and females. What is the term for a gene that has multiple forms or variations at a single locus? A. Haplotype. B. Allele. C. Locus. D. Intron. The correct answer is B. An allele is one of several alternative forms of a gene that can occur at a specific locus on a chromosome. Which of the following techniques is commonly used to amplify DNA segments for genetic analysis? A. Western blotting. B. Gel electrophoresis. C. Polymerase chain reaction, PCR. D. Microarray analysis. The correct answer is C. Polymerase chain reaction, PCR, is a technique used to amplify specific DNA segments, enabling detailed genetic analysis. Which gene mutation is most frequently associated with an increased risk of breast cancer? A. CFTR. B. BRCA8. C. SOD8. D. MYHSAT. The correct answer is B. Mutations in the BRCA1 gene significantly increase the risk of developing breast and ovarian cancers. Which process is defined by the exchange of genetic material between homologous chromosomes during meiosis? A. Transduction. B. Crossing over. C. Transformation. D. Splicing. The correct answer is B. Crossing over is the process where homologous chromosomes exchange segments, promoting genetic variation in offspring. Which of the following describes the consequence of a frameshift mutation in a gene's coding sequence? A. Inserts a premature stop codon. B. Shifts the reading frame, altering all downstream amino acids. C. Causes a single amino acid substitution. D. Has no effect on protein function. The correct answer is B. A frameshift mutation changes the reading frame, leading to entirely different amino acid sequences downstream, often resulting in a non-functional protein. What is the main purpose of using a karyotype in genetic studies? A. To identify single nucleotide polymorphisms. B. To visualize the number and structure of chromosomes. C. To measure gene expression levels. D. To analyze mitochondrial DNA variations. The correct answer is B. A karyotype displays the chromosomes of an organism, allowing for the detection of chromosomal abnormalities in number or structure. In a pedigree analysis, which inheritance pattern would display affected individuals in every generation and affect both males and females equally? A. X-linked recessive. B. Autosomal recessive. C. Autosomal dominant. D. Y-linked. The correct answer is C. An autosomal dominant inheritance pattern shows affected individuals in each generation and affects both genders equally, as it is not linked to sex chromosomes. Which term best describes the condition when an individual has 2 different alleles at a single genetic locus? A. Homozygous. B. Polygenic. C. codominant. D. Heterozygous. The correct answer is D. Heterozygous refers to having two different alleles at a specific locus on homologous chromosomes. How do geneticists differentiate between a mutation in a coding region versus a regulatory region of a gene? A. Mutations in coding regions affect gene expression patterns. B. Mutations in regulatory regions impact transcription levels without altering protein structure. C. Only coding region mutations are inheritable. D. Regulatory region mutations produce truncated proteins. The correct answer is B. Regulatory region mutations influence the levels of transcription but do not directly change the protein structure encoded by the gene. Which of the following diseases is commonly associated with a trinucleotide repeat expansion mutation? A. Cystic fibrosis. B. Huntington's disease. C. Sickle cell anemia. D. Hemophilia. The correct answer is B. Huntington's disease is caused by a trinucleotide repeat expansion, where excessive repeats lead to protein malfunction and neuronal damage. In which phase of cell division do homologous chromosomes segregate, leading to a reduction in chromosome number by half? A. Mitosis. B. Meiosis I. C. Meiosis II. D. S phase. The correct answer is B. During meiosis I, homologous chromosomes separate, reducing the chromosome number by half, which is essential for producing haploid gametes. What is the role of histones in the structure of human DNA? A. They repair DNA damage. B. They unwind the DNA during replication. C. They package DNA into chromatin. D. They catalyze DNA synthesis. The correct answer is C. Histones are proteins that help organize and compact DNA into chromatin, enabling efficient packaging within the cell nucleus. Which of the following describes a polygenic trait? A. A trait determined by a single gene with multiple alleles. B. A trait influenced by multiple genes and environmental factors. C. A trait passed only through mitochondrial DNA. D. A trait that skips generations due to recessive inheritance. The correct answer is B. Polygenic traits are influenced by multiple genes and environmental factors, such as height or skin color, contributing to a continuous range of phenotypes. Which of the following best explains the function of RNA splicing in human cells? A. It removes introns from pre-mRNA, enabling proper translation. B. It adds a 5' to RNA transcripts. C. It synthesizes tRNA for translation. D. It edits mRNA sequences post-transcription. The correct answer is A. RNA splicing removes non-coding introns from pre-mRNA, producing mature mRNA that can be translated into proteins. How does a mutation in a promoter region of a gene likely affect that gene's expression? A. Increases the protein length. B. Alters the amino acid sequence. C. Reduces or abolishes gene transcription. D. Causes frameshift mutations. The correct answer is C. A mutation in a promoter region can prevent binding of transcription factors, reducing or stopping transcription of the associated gene. What characteristic defines a linkage disequilibrium, LD, in human genetics? A. Independent assortment of alleles at different loci. B. Non-random association of alleles at two or more loci. C. Mutations confined to mitochondrial DNA. D. Chromosome segregation errors. The correct answer is B. Linkage disequilibrium occurs when alleles at different loci are inherited together more frequently than expected by chance. What effect does a non-synonymous mutation have on a protein? A. Introduces a stop codon. B. Has no effect on the amino acid sequence. C. Changes the amino acid sequence of the protein. D. Deletes an amino acid from the protein. The correct answer is C. Non-synonymous mutations alter the codon, resulting in a different amino acid being incorporated into the protein sequence, potentially affecting protein function. Which of the following mechanisms allows for the generation of antibody diversity in the immune system? A. Alternative splicing. B. Somatic recombination. C. Point mutation. D. DNA methylation. The correct answer is B. Somatic recombination, which occurs in B cells, generates a vast diversity of antibodies by rearranging gene segments. Which of the following is the primary function of the p53 protein in human cells? A. To repair DNA damage. B. To facilitate cell division. C. To induce apoptosis in response to DNA damage. D. To promote telomere elongation. The correct answer is C. The p53 protein plays a crucial role in inducing apoptosis in cells with DNA damage, preventing the propagation of potentially cancerous cells. Which of the following processes is responsible for creating genetic diversity through the exchange of genetic material between homologous chromosomes? A. Transcription. B. Recombination. C. DNA replication. D. Translation. The correct answer is B. Genetic recombination, occurring during meiosis, involves the exchange of genetic material between homologous chromosomes, contributing to genetic diversity. Which genetic phenomenon explains why certain genetic traits are inherited together more frequently than expected by independent assortment? A. Genetic drift. B. Linkage. C. Mosaicism. D. Codominance. The correct answer is B. Linkage refers to the tendency of genes located close to each other on the same chromosome to be inherited together, contrary to Mendel's law of independent assortment. In human cells, which of the following enzymes is crucial for preventing DNA from becoming overly supercoiled during replication? A. DNA ligase. B. Helicase. C. Topoisomerase. D. DNA polymerase. The correct answer is C. Topoisomerase prevents DNA from becoming overly supercoiled by creating temporary cuts in the DNA, which relieves tension during replication. Which term describes the phenomenon where a single gene influences multiple phenotypic traits in an organism? A. Epistasis. B. Polygenic inheritance. C. Pleiotropy. D. Codominance. The correct answer is C. Pleiotropy occurs when a single gene affects multiple phenotypic traits, as seen in disorders like Marfan syndrome. A silent mutation in a gene sequence is one that A. Does not alter the amino acid sequence of the resulting protein. B. Results in a frameshift mutation. C. Introduces a stop codon. D. Changes protein function significantly. The correct answer is A. Silent mutations do not change the amino acid sequence due to redundancy in the genetic code, often having no functional impact on the protein. Which of the following genetic terms best describes an observable trait expressed by an organism? A. Allele. B. Genotype. C. Locus. D. Phenotype. The correct answer is D. Phenotype refers to the observable characteristics of an organism, influenced by both genetic makeup, genotype, and environmental factors. Which type of chromosomal mutation results from a portion of one chromosome being inserted into a non-homologous chromosome? A. Duplication. B. Inversion. C. Translocation. D. Deletion. The correct answer is C. A translocation mutation involves the transfer of a chromosome segment to a non-homologous chromosome, potentially disrupting gene function. Which enzyme is responsible for synthesizing the RNA primer during DNA replication in human cells? A. RNA polymerase. B. Primase. C. DNA ligase. D. Telomerase. The correct answer is B. Primase synthesizes a short RNA primer that DNA polymerase can extend, enabling DNA replication to initiate on the lagging strand. What is the consequence of a nonsense mutation within the coding sequence of a gene? A. It changes one amino acid in the protein. B. It has no impact on the protein. C. It introduces a premature stop codon. D. It causes a frameshift mutation. The correct answer is C. A nonsense mutation introduces a premature stop codon, often resulting in a truncated, non-functional protein. Which of the following is a key difference between somatic mutations and germline mutations? A. Somatic mutations are inheritable. B. Germline mutations only affect somatic cells. C. Somatic mutations cannot be passed to offspring. D. Germline mutations occur only in liver cells. The correct answer is C. Somatic mutations occur in non-reproductive cells and cannot be passed to offspring, while germline mutations, occurring in gametes, can be inherited. Which term describes the occurrence of different forms of a gene at a specific locus within a population? A. Phenotypic variation. B. Allelic frequency. C. Polyploidy. D. Allelic variation. The correct answer is D. Allelic variation refers to the existence of different alleles at a locus, contributing to genetic diversity within a population. Which molecular process results in the formation of a pre-mRNA transcript from a DNA template? A. Replication. B. Translation. C. Splicing. D. Transcription. The correct answer is D. Transcription is the process by which RNA polymerase synthesizes a complementary pre-mRNA strand from a DNA template. Which of the following processes helps prevent shortening of chromosomes in rapidly dividing cells? A. Transcription. B. Telomerase activity. C. DNA polymerase activity. D. Primase synthesis. The correct answer is B. Telomerase adds repetitive nucleotide sequences to the ends of chromosomes, preventing telomere shortening in dividing cells. The term penetrance in genetics refers to A. The likelihood of a gene being expressed. B. The intensity of the gene expression. C. The mutation rate of a gene. D. The ratio of dominant to recessive alleles. The correct answer is A. Penetrance is the probability that a person carrying a specific genotype will exhibit the associated phenotype.